Good afternoon and welcome to my MCC Scholars Day talk. Let me get my PowerPoint going here and then we can begin. Yeah, share. That's what I meant. Sharing is caring. All right. I am Professor Stevens, Rich Stevens from the biology department, and I have been doing some research for the past few years with my colleagues, students, and children on the cutaneous bacteria of spotted salamanders right here in Western New York. Spotted salamanders are fairly common in this neck of the woods throughout the eastern United States, all the way down to Georgia uh, in the eastern US and up into Canada, Maine, in southern Canada. And anyway, they are extraordinarily beautiful. As you can see, these are some pictures I took right on my cell phone. And they spend much of the year, they're members of the Ambystomid family, which is the mole salamander. So like moles, they dig underground, spend most of the year underground eating bugs and sleeping and doing their business. But on warm, rainy nights, they walk around on the surface of the forest, and especially the first really warm, rainy night of spring, they wake up from sleep. Brumation is the technical term. I'm trying to avoid being too technical. It's like the amphibian version of hibernation. But they wake up and they emerge and they walk down to their breeding ponds. And around here, in some places, you can see them by the dozens. In other places, by the hundreds. Or a friend of mine lives a little farther south down near Honeoye, and he has thousands that he can see over the course of the first few warm, rainy nights in the springtime. So salamanders they are amphibians. And the other amphibians that you're familiar with are frogs and toads. And Sicilians that you can see here, they are tropical creatures that you will not see in this neck of the woods. They look rather snake-like and worm-like, and that body plan of leglessness has evolved multiple times. There's lizards that are legless, Sicilians that are legless, of course, snakes are legless. So anyway, what separates amphibians from other animals that are sort of similar, like reptiles? Salamanders look a little bit, I mean, if you look at that spotted salamander, it's sort of got a lizard body plan. But what makes it different is amphibians, and I actually have an amphibian here. I'm going to try not to scare him, but this is, oops, I wanted to stop my screen share. Don't jump, Rubius. Rubius is a Malayan leaf frog, and I wish you could touch him, but he's got very thin and sort of slimy skin. He doesn't have skin like me. Reptiles have, well, I'll say humans have reptile skin. Reptiles don't have human skin, but I have dry skin, so I can go out on a hot sunny day and not shrivel up like a raisin. But not Rubius here. Rubius does not go out on hot sunny days because his skin is not waterproof. His water would all evaporate, and he would do like the earthworm do on a hot sunny day. They shrivel up and they die. So amphibians have thin, moist, glandular skin that I'll show you here in just a minute, whereas reptiles have very dry and waterproof skin. So that's why reptiles do well in hot, dry climates. Amphibians really like the moist, the rainforests, and just generally wet, moist habitats. Rubius, I'll put you back. All right, I'm going to pause this presentation for a minute and put Rubius back in his tank, and then we will proceed. All right, and we are back. Let's get back to the PowerPoint. So amphibians, moist glandular skin, and when they lay eggs, their eggs are like their skin. They're not waterproof, so they can't do like chickens do and lay their eggs in a nest, just right in the barn where they're in the air. Most amphibians lay their eggs in the water, like literally. That's why these salamanders move to the water to breed. So these guys have been around. I heard somebody once call them lower vertebrates, but I wouldn't call anything a lower vertebrate, maybe except me. But they have been around doing their thing, surviving since they crawled out of the swamp 380 million years ago. That is an exceptionally long time. They are excellent, amazing survivors, but they're having a hard time today. Amphibians have, like I said, moist, slimy-like glandular skin. You can sort of see how that spotted salamander looks slimy. And they use their skin's been called Swiss Army skin. It's multi-purpose. They don't just use their skin like you do for protection against pathogens, viruses, and bacteria and fungi, although they do that too. They have poison glands and they use their skin to deter predators. Amphibians do not drink in the classic sense. Water doesn't go in their mouth hole. They absorb water through their skin. So Rubius, when he's thirsty, he just literally lays in a puddle of water and absorbs water by osmosis. Good term for you biology students. And they also use their skin for gas exchange. Many amphibians have no lungs or no gills, and they just do gas exchange through their skin, and that's not something you can do through your skin. And last but not least, ion regulation, electrolyte balance, sodium, potassium, stuff like that. So their skin is super important to them. And now there is a fungal pathogen, one of what's called emerging fungal pathogens. This was discovered in 1999. And at a scientific meeting, which is very important, administrators and deans send your faculty to scientific meetings. It was a scientific meeting in 1999 or 98 when several different herpetologists, people that study amphibians and reptiles, were talking about how all their frogs had died. And what they eventually found was in 1999, they discovered a fungus called the chytrid fungus. Batrachochytrium dendrobatitis is its scientific name. But it destroys that special amphibian skin. It basically eats away at their skin, which means no protection against pathogens. They can't drink, can't regulate their ion, their electrolytes. And as you can see here, it's been called, the frog apocalypse. It affects salamanders too. But thanks to the chytrid fungus and habitat loss and pollution and pesticides and a host of other things, global climate change. 40% of amphibian species are threatened or endangered. They are the most endangered as a group of vertebrates, animals with backbones. So this chytrid fungus is a big cause of that. There's a new kid on the block, B. sal, which is of Asian origin, like BD is, but it moved into Europe, where the salamanders of Europe have no immunity to it, and it's been decimating them. And the concern is North America, the United States especially, from Virginia on south, the southern Appalachians, is a biodiversity hotspot for salamanders. The United States Tennessee has more salamander species than Europe and Asia combined. So it's amazing, the biodiversity. I went to graduate school. I lived for Tennessee. I lived in Tennessee. I didn't live for Tennessee. I lived for myself. I lived in Tennessee for 10 years, and some days in the southern Appalachians after a warm rain, you literally just I had to watch where I put my foot every step. There was just so many salamanders, just dozens and dozens and dozens of species of salamanders. So the concern is when B. sal, and it's probably not a matter of if, but when B. sal comes to North America, it's going to decimate our salamanders. And so there have been some lab research studies that have sort of tested how are our native North American species, how do they do with B. sal? And the answer is overall not well at all, except spotted salamanders. Spotted salamanders seem to have some innate immunity to this pathogen, this fungal disease, but B-cell is a concern. And here's the deal: cutaneous bacteria, bacteria that live on the skin, and the average amphibian has hundreds of species of bacteria that live on their skin. It's called the cutaneous microbiome, or the dermosphere. These bacteria make metabolites that are strongly antifungal. This toad you see here is a boreal toad. They were extirpated in many places in Colorado due to the BD fungus. And what they found was that one particular type of bacteria, which we'll talk about in a minute, is so strongly antifungal that they literally just dipped these toads in this bacteria and then released them into the wild. And that bacteria gave them the power to survive in environments with BD. And we'll talk more about that. But cutaneous bacteria make chemicals that are antifungal for a variety of reasons. They do that, but that's very important to amphibians, and there's been a lot of research. BD is here in Western New York. It's here all over. It's what's called enzootic. It's almost like the common cold. It's just around all the time. Sometimes you get sick from it, sometimes you don't, but it's just there 24/7, 365. And BD is like that right here. All right, so the purpose of this study that we started with a couple of independent students, Professor Tim Tatakis, myself, Suzanne Long, and our independent study students, Kristen Fellows, and my daughter, Annie Stevens. We started this 3-4 years ago, and we just wanted to identify as many species, figure out what species were on spotted salamanders, because a bunch of studies have looked at frogs. Few studies have looked at salamanders, but nobody, and I mean nobody, had looked at cutaneous bacteria in these mole salamanders, and especially the spotted salamander. It was just nobody done it. So we decided that was where we fit in. That was what we were going to do. So we wanted to identify as many as we could. So they are relatively easy to capture if you know where to go, and I'm not going to tell you where to go, but if you know, on the first warm rainy night in the middle to late March, usually, you can go out with your rain gear and your boots, and you can find dozens of them. So we would just capture them by hand with gloved hands. We would rinse them to rinse any bacteria that's sort of from leaves or dirt that were on their skin. And then we would take a sterile swab, like just a long Q-tip, basically, that was sterile, and we would rub it all over the salamander, and then we would rub it onto a Petri dish and incubate those Petri dishes with R2A agar for about 48, 72 hours at room temperature. We didn't need to keep them in an incubator because these were on amphibians that were roaming around at, 12, 15, 16 degrees Celsius. No need to incubate them at 37 degrees Celsius. And we saw what we grew. And larvae, so again, they go to the water, they lay their eggs in the water, and they have a tadpole-like, a frog-like life cycle. The egg hatches into a larva that has external gills. Sorry, I can't say external gills without doing that any more than I can talk to my students about deer antlers without doing that. So they have external gills that tadpoles keep their gills inside, but salamanders, that's how you can, if you're ever wondering, how do you tell a tadpole, which is a larval frog, from a salamander, put them in a beaker of non-tap water, and salamanders, you'll be able to see their gills like you can hopefully see on this little guy right here. So those are his gills. They look kind of like, if you know what an axolotl is, I have an axolotl right behind this computer, but she is not, she's hiding in her little hidey hole. But we would capture larvae by just dip netting. So just sticking a net in, lifting it up, and looking for them. And we would swab them as well. And then what we would do is we would take, so if we go back to my Petri dish here, we would take that as a colony. So each bacterial colony, each species looks a little different. So I would scoop a little bit of that up, put it on a fresh Petri dish that's called subculturing. So this is a subculture right here. We'd subculture, get pure colonies, and then I would grow them in liquid nutrient broth for 24 hours, and then I would spin it down in a centrifuge, freeze it, send it to a lab where they would sequence the entire 1.5 kilobases of the 16S ribosomal RNA gene. And that is a fantastic way of identifying what species and strain of bacteria you have. So there's some cell pellets that were about ready to be shipped off to the lab. And then so I took the DNA sequences that the lab sent and used software from the National Institutes of Health free. You can just Google BLAST. It has this fantastically wonderful, super easy to use website. You just upload your DNA sequence and it tells you exactly what species of bacteria you have. It was joyous to use, loved it. So here's sort of our numbers. We sampled quite a few more adults than this, but at 16 adults over 2 years, we actually sent bacteria to the lab to be identified. We had 3 juveniles. Juveniles were hard to catch. Larvae were really hard to catch, especially last year. And a metamorph is right after they switch from their larval stage, they lose those external gills, they grow lungs, grow a tail. Well, they grow a tail, they had a tail, but their legs emerge and now they're ready to crawl out of the swamp on land. I had been looking for, like, right at that moment, right after they move onto land, could I catch one? So I went out one night and I was looking for juveniles, and a bunch of mountain bikers came down the trail of my study site. And, there was like 15 of them. And I was like, oh, Jesus. I was not pleased, but I got off the trail so these weekend warriors could run by. And while I was waiting for them to go, they were actually very polite and thanked me for getting off the trail. I'm just a grouch. I flipped over a log. There was my metamorph. So hats off to the mountain bikers. Thanks, fellas and ladies. I found the metamorph I was looking for. So happy. Okay, so here is some of the results finally. Xanthinobacterium lividum. This grows beautiful purple colonies that you can see there due to a metabolite that the bacteria make called violacin. And violacin is probably the most antifungal chemical known. And it was this bacteria, Xanthinobacterium lividum, or J-liv, they were the ones that I was telling you about that were inoculated onto the toads in Colorado before they were reintroduced. We found quite a bit of J-liv in 2019. Over 50% of the salamanders had this bacteria on them. This past year, the metamorph had it, and one of my 11 adults had it. So it was a little less common this year, but it is powerfully antifungal, antibacterial, anti-tumor, and just a really neat. And I loved every time I would swab a plate, I would just look to see if that beautiful purple colony was there. So Xanthinobacterium was pretty common on our salamanders. Pseudomonas is a genus of bacteria that we found quite a few species. I'll show you some more in the next slide. But Pseudomonas fluorescens is another strongly antifungal bacteria. It makes chemicals. It makes many different chemicals. I sort of, did some internet research on what are the antifungal metabolites, and there are many. But this species is so strongly antifungal that it's used in aquaculture. So when they're like growing tilapia or keeping fish for hobby, they put this bacteria in the water to keep fungi away. So very strongly antifungal. If you do some internet research on this, this is a classic antifungal. It was found in 2 adults this year. Here's some of the other Pseudomonas species. A stands for adult, M was the metamorph, J for juvenile, but Pseudomonas as a genus is pretty strongly antifungal. And a shout out to my independent study MCC students. Kristen Fellows, and my daughter Annie, because Pseudomonas aeruginosa, that is the first species that we identified with the help of Professor Long using biochemical techniques. And that really got me personally just addicted to this research. And I'm not a microbiologist at all. I'm really a zoologist, a herpetologist, but oh, I have fallen in love with bacteria. So if you're watching this, Professor Long, thank you for what you did. And to our students, Chrysiobacterium, this is another species that has been found in frogs and salamanders, and it's been used sort of in tests. When they put this bacteria against the chytrid fungus, it is strongly antifungal and it stops the BD fungus. So it is an excellent bacteria that we found on the metamorph. Yeah. Serratia liquefaciens. This is something that you Bio 156 students actually use in the lab. It makes a metabolite called chitinase. And ACE is an enzyme. Chitin is the material that makes up the cell wall of fungi. So this metabolite from this bacteria is antifungal by breaking down the cell wall that the fungi have. Okay, and many species of Bacillus were found. Interestingly, if you look at the age classes, and excuse my sinusitis, juvenile, larva, metamorph, bacilli were not found. Bacillus, the genus, different from bacilli, shame on me. But anyway, bacillus species were not found in the adults, and we only looked at a handful. So I didn't do a complete survey. This is not a complete list of what's on spotted salamanders, but as much as we did, it's sort of surprising. And it just gives you a little hint that maybe the species of bacteria change over the lifespan of the salamander. Pretty intriguing. All right, let me wrap it up here, if my technology will work. There's a beautiful little juvenile from last fall. A total of 33 species were identified. I just had the second bit of this accepted for publication. So if you would like a whole list, I can send you a copy of the paper. It's going to be published in June in Herpetological Review. So again, 33 species were identified over the course of this study, but that's a drop in the bucket. There's many more. And so we've identified specific antifungal bacteria. But studies have sort of the current thinking over the past few years is, in spite of J-Liv being very successful at helping these toads survive, it really is the community and that it's what's called an emergent property. So as all those bacteria and all their metabolites, that's really what's giving them the antifungal capacity. But we have disrupted something, dysbiosis going on, that has allowed these fungal diseases to emerge. And there's fungal diseases in snakes, in bats, in many different organisms, as well as the BD and B cell. And we have a very small sample size, but our research does hint that maybe the bacteria are changing. The community, the species are changing due to habitat changes, physiological changes, something that's going on. And certainly habitat changes because larvae obviously are aquatic, whereas adults are mainly terrestrial. Okay, and I am indeed very grateful for so many things and so many people, but my son and daughter, Annie and Max, former and current MCC students, respectively, they helped me every bit of the way. I never was in the field ever without one or both of these. For most of it, it was both. They are always up for going and doing field work, and I love them for that and for much, much more. Adam Taylor was one of my more recent independent study students. Shame on me for not putting Kristen Fellows here, who was my first independent study student. So Kristen Fellows, thank you for all you did for this work. The MCC Foundation has been excellent and has funded our research for 2 years, and I thank them for that. Our wonderful Department of Biology, James Murphy, has been so supportive in so many ways. Jill McPhee, Suzanne Long, and Charles Wertzer helped me with the lab microbiology skills that I am so woefully lacking in before I started this, and I consult them regularly for help. Margaret Kaminsky is the best dean ever, and she gave financial and moral support throughout this research, excellent human being, and thanks to the Department of um, Environmental Conservation here in New York State for giving me a permit and allowing me to go do this to our salamanders and for helping us support these critters. So thank you for listening, and everybody, as they say, have a great day.